Good afternoon to everybody and welcome to Valencia, where the biomarkers meeting in reproductive medicine will be held over next 2 days. Okay, and we will start the biomarker, the workshop, the trophectoderm biopsy workshop. Okay, and let me first of all introduce myself. Thank you. Okay, I am Dr. María Eugenia Póo. I am biologist, graduate from the University Simón Bolívar from Caracas, Venezuela, and I work for many years in leading IVF programs in Latin America and Spain in EV Valencia, and share with the Carlos Simón research team and work in the National Stem Cell Bank deriving human embryonic stem cells deriving from single blastomere without embryo destroying. Okay? Nowadays, we join with Igenomix from last 4 years, and I am the embryology director, giving support worldwide to IVF clinics to establish PGS or PGD program in the IVF labs. And we have a lot of experience establishing good culture conditions in order to improve the PGS program in different labs, okay? And then we start the workshop, just showing you the learning objectives of this workshop. First of all is to understand what is the Preimplantation Genetic Diagnosis, to learn about the evolution and the future of Preimplantation Genetic Diagnosis, to learn the best strategy for trophectoderm biopsy, to update the IVF lab culture conditions for PGS, and to discuss how to improve the PGS outcome with vitrification. Okay, let me to open the presentation. Let me. Okay. We start talking about the evolution and the future of Preimplantation Genetic Diagnosis. Recently, last year, commemorate the 25 years of Preimplantation Genetics Diagnosis, and really have been 25 years of changing lives, okay? And if we make an overview of history of PGD, I will tell you that in 1968 was performed the first sex determination in animal model, specific in rabbit embryos at Blastocyst stage. In 1978, as you know, birthed the first IVF baby, and in 1990, the first PGD baby born. In 1993, the technology of fluorescent hybridization in situ hybridization analysis was used for sexing and also for PGD aneuploidies. In 1996, this same technology was used for chromosomal translocation, and in 1998, also used for late-onset common disorder with genetic predisposition. And in 2000, start to use for HLA typing, and in 2002 arrived to the first 1,000 babies, PGD babies, okay? And in 2009, after a long time using FISH technology, finally arrived a new molecular technology called CGH Arise, and finally in science 2 years ago, the NGS, or next generation sequencing, arrived to perform the preimplantation and aneuploidy testing. During a long time, the FISH technology was used in order to perform PGD, mainly at the beginning to apply it in patient carrier for monogenetic disease, and mainly sexing embryos and transfer, and especially for X-linked disease, and choosing only female embryos for transfer. And when start to apply for screening to aneuploidies, a lot of publication was telling and not recommend anymore to perform the PGD analysis, because really they don't observe any improvement in clinical outcomes. And other groups, of course, show performing a randomized clinical trial in patients with recurrent implantation failure and also for the advanced maternal age, demonstrate that really we can improve the pregnancy rate, ongoing implantation rate, and live birth rates for both groups, for recurrent implantation failure and for advanced maternal age. And another groups telling us about the probably the problem with the technology is the patient inclusion criteria, the embryo biopsy procedures, and the embryo culture condition, and the type of genetics analysis performing, and probably with for this reason, the results are not really good. Finally, in 2009 arrived the new molecular technology, and now the PGD is called PGS for screening aneuploidies, okay? Preimplantation Genetic Screening. And PGD is only used for monogenetic disease, single gene disorder. And this is a powerful tool in order to assessment the embryo viability, improving the implantation, decreasing the miscarriage rates, and decreasing the risk of abnormal offsprings. And it's true, and we know from different randomized clinical trials from many groups from US, that this kind of technology can be applied in any patient with good prognosis patient, even in the first IVF cycle with no prior history of miscarriage. But sometimes happens that if in your country have some legal frame that you need to apply, you need to have an indication to apply this kind of technology. And this is the situation in Spain, for example. We belong to the ESRE consortium, and for applying the PGS in the patient, we need to include the patient under some indication. And the main indication are advanced maternal age, over 30 years, prior pregnancy or child chromosomically abnormal, multiple implantation failure, 2 or more, recurrent miscarriage, and severe male factor, only considering low sperm count. And we know that trisomic embryos reach a blastocyst stage with similar frequency than normal embryos. And in patients over 30 years old, okay, you can see here, in older patient, the rate of blastocyst is identical for normal embryos and for trisomical embryos. And that means that the morphology of the embryo is not anymore the best criteria to select the embryo for transfer. And nowadays, applying for many years the PGS in our centers, we know and we see a lot of good blastocysts. In this case, it's a euploid blastocyst. But for example, this is a monosomy. This is an embryo carrier of monosomy 12. And as you can see, looks very nice blastocyst. And then only the morphological criteria fails selecting the best embryo, and the transfer of good morphology embryos not always means good quality embryo. Very briefly, I would like to mention the workflow of array technology. And at the IVF lab, you perform the embryo biopsy at day 3 or at blastocyst stage. And the biopsy cells you put inside the PCR tubes and then send to the genetic lab. There, start with the whole genome amplification DNA, label with the fluorochrome in red or blue, and precipitate the DNA, perform the hybridization, wash, and scanning, and include this slide in a scanner using a special software, and you can obtain this kind of profiles. And each column represents a chromosome from 1, 2, 3, and so on until 22, S and Y. And you have in each chromosome more or less between 65 to 70 points of hybridization, okay? And you have a threshold, and if all the points maintain in the threshold, this is considered normal. And in this case, it's a euploid embryo. In this case, you can see that the chromosome 13 is lower to the threshold, and then this is a loss of this chromosome. And you can identify also partial gain or partial loss. And for example, here you can see the gain of 21, and in many cases you will observe aneuploided embryos with many aneuploidies in the same embryo. Okay, and you can also see embryos named a complex abnormal, that all the chromosomes are completely aberrant. And if now we compare the clinical parameters, like pregnancy rate per transfer or implantation rate, comparing PGS versus FISH, you can see how the technology improve the clinical outcomes, okay? And is higher in the case of older patients. Many reports nowadays is available in the peer-reviewed journals. And for example, in this case, using trophectoderm biopsy, vitrify the blastocyst, and then perform the analysis and transfer in subsequent cycle. And in this case, all patients have a normal ovarian response, and a single embryo transfer of 100 frozen blastocysts. And you can see here the clinical, the rate of pregnancy, the pregnancy rate was 73%, and the abortion was almost 3%, and the implantation rate was almost 65%. And another publication, another group, demonstrate that you can, even using fresh transfer, really you can get benefit from PGS, okay? This is a randomized pilot study, and you can see here a fresh transfer, frozen transfer, fresh transfer without analysis, frozen transfer without analysis, and fresh transfer without analysis and fresh transfer with analysis. And then you can see how you improve the pregnancy rate in the group that you make the analysis for aneuploidy. Is almost 71% comparing to 46%. And another study telling us about the strategy of vitrificate the blastocyst and transfer in the subsequent cycle in order to facilitate the elective transfer for infertile women with advanced maternal age. And in this case, you have a fresh transfer without analysis, a frozen transfer without analysis, and frozen transfer with analysis. And then you compare the implantation rate, how increase, okay? And the most important thing is how decrease the abortion, okay? And the ongoing pregnancy rate increased significantly, okay? Another study also performing fresh embryo transfer using different patients with under different indication, male factor, tubal factor, and in all of case, this is the average of age, 32 years old for both study groups, and you have see here that in both groups, more or less, you have the same amount of blastocyst, and here you can see how you can improve the clinical implantation, and also you can see here that the embryos selected after comprehensive chromosome screening are also statistically significantly more likely to progress to delivery than unscreened embryos, okay? And finally, 2 years ago arrived the NGS, the next sequence generation sequencing, and there are several commercially available platforms, and one of them, and is the most famous, Illumina, okay? And the protocol basically starts with the amplification of whole genome amplification. You perform the tagmentation and label the fragments with a barcode, prepare the library, and then start the sequencing and matching with the with a reference DNA. And of course, start appearing the publication demonstrating that the new technology NGS is we can obtain similar or better results comparing to arrays. And in this case, the group of Francesco Fiorentino from Italy demonstrate that the performing day 3 biopsy, you can obtain good results. And also our group demonstrate using the Illumina platform that we can study and identify segmented aneuploidy in the same concordance rate that using array technology. In that case, you can see here both studies from Fiorentino and our group, and comparing for whole chromosome aneuploidies, we have a concordance of 100% if we compare CGHRI with NGS. And for segmental aneuploidies, some professionals in the field suspect that the segmental aneuploidies are really artifacts and not are through aberrations, chromosomal aberrations. And in our group, really believe in segmental aneuploidies, and we perform a study with a total sample 124 and divide in 2 groups, the control groups and couples carrier of translocation, And the study group is couples with normal karyotypes. And comparing in the control groups, comparing CGH rate between CGH rate and NGS, the concordance was the 100%. And in the study group, identifying new events of segmental aneuploidies, the concordance was 97.7%. And the average of concordance was 98.4%. And really to demonstrate that the segmental aneuploidies is not artifact, we fixate whole blastocysts and identify the segmental aneuploidy using some proofs to identify the aberration, and in 96% of the segmental events was detected and confirmed by FISH. Okay, another platform commercially available is the Live platform for Thermo Fisher, and in this case is completely different from Illumina. And in this case, you have a chip, a conductor chip, and every time that some nucleotide is incorporated in the sequencing, the pH in the chip change and emit a signal, and then perform the sequencing, recording the change of the pH. Okay, in Illumina case, each base has a fluorochrome and emit a light and recognize which base incorporate in the sequencing. And of course, we should to demonstrate that using this platform, we have the same results comparing with array. This is a case for euploid embryos. And this is another case for an euploided embryo. You can see here a monosomy 15 and a trisomy 19 and monosomy 21. And it was also demonstrated in using the PGM from Life Technology. And there is a lot of publication demonstrating that the clinical results performing NGS are similar that using array nowadays. And you can see here for a patient advanced maternal age, the pregnancy rate per transfer was almost 64%. And you can see here for young, good prognosis patients, you can see here NGS arrays, SNPs arrays, and CGS arrays. And you can see here the pregnancy rate per embryo transfer. And evidence is not statistical significant, but looks that probably the CGH array is better than the other technology. But remember that really the new technology NGS appeared in 2013. And during these two last year, really we are in the learning curve trying to identify or trying to interpret the profiles. And probably we at the beginning overdiagnosis the embryos, and we suspect some abnormalities, or maybe it's normal, but you cannot take a risk to say it's normal, and then you prefer to say abnormal. And the same happened when started 5 years ago with arrays. When you start and when you learn, and you finally know how the interpretation of the profiles, probably you obtain better results. And this is a randomized study appeared last year, okay, and comparing NGS with array, and finally we can obtain similar clinical data, and seems that NGS could be better, okay, for the diagnosis, and probably the resolution, and probably we can identify more different events. Some authors mention about the mosaic things. We can identify more events that can improve the clinical results. And another approach that is not very expanded nowadays is the analysis of blastocell or the blastocyst fluid. And basically, this fluid is obtained using an ICSI pipette and aspirate the fluid inside the cavity. And you need to avoid, it's a little tricky technique, okay? You aspirate the liquid, and you need to avoid any cell inside. And in this first study, can you see here the concordance, the karyotypes comparing the karyotype in the liquid of blastocell comparing with the karyotype of the blastocyst is not really good. The concordance is 50%. Okay, probably this approach will be must be improved, and indeed. Last month was published in Fertility and Sterility by the Christina Magli. Really better result comparing the results, biopsy, polar bodies, blastomere, trophectoderm cells, or blastocellic fluid. And really we can see here very good results and a very high concordance in ploidy and very high concordance in chromosome. And probably could be a new way to determine the genetic status of the embryo. But the gold standard for the future approach is really to apply non-invasive PGS. At the end, the biopsy procedure is invasive procedure, and really the embryologists need to have very good skills, high experience to have really good results. And the idea is in the future, maybe I don't know how many years takes to arrive to the final demonstration that the analysis of the culture media, the DNA contained in the culture media, will be really representative of the genetic status from the embryo. We start with some projects at this moment, but we have surprising that we identify non-human DNA in culture media. And probably we need to start to learn how to work in the IVF labs because probably you cannot see any bacteria, any fungi with your eyes and under the stereoscope, but in the culture media you have DNA from microorganisms. And in order to apply this technology, we need to be sure that the culture condition will be perfect. And only the DNA contained in the embryo culture media is human DNA, and human DNA from the embryo, not from the embryologist. It's an also very good point, okay? And it's a very good point also using the other technologies. But when you put the cell into the tubes, you need to take a lot of care to avoid any extra contamination from external DNA. For example, if when we receive a case, many embryos, maybe 8 embryos, and the results are completely normal and all the embryos have the same gender, this is not possible. And probably you suspect some contamination. And it is very, very important how to work in the IVF lab to get really good result, and really the result is representative of the genetic status of the embryo. Another good point is when perform the biopsy, day 3, day 5, until maybe 5 years ago, most of the labs performed day 3 biopsy. Some groups arguing that the just one cell is not really representative from the rest of the embryo. And sometimes happen, or some people talking about the mosaicism, and another groups are talking about the self-correction of the some abnormalities between day 3 and day 5. And nowadays, most of the groups are moving to perform the biopsy at day 5. And there is many reasons to perform the biopsy at day 5. One of them is you take more than one cell, and the number of cells not should be 3, for example, because probably you kill 2 of them with the laser shot, and finally you analyze only 1. In that case, it's better that you perform the biopsy on day 3. And when you perform the biopsy on day 5, it's recommended to take minimal 6 cells, between 6 to 8, 10 cells. And this is very important, and probably Nuno in the next talk explain very good all the approach or all the methodology that you need to use to get very good sample in order to get the advantage, the really advantages comparing day 3. In our group, we have a lot of experience performing biopsy on day 3. And the problem in our group is that more than 80% of our patients belong to advanced maternal age group, are older than 40 years old, and it's very difficult. Most of them are low responder, and probably you have a few embryos for analysis. And probably if you have only 2 embryos on day 3 and you culture until day 5, probably not arrive to day 5, and not arrive to day 5 not but because you have problems in the culture. Maybe you have problem in the culture, but if you have very good standard culture condition in your lab, probably the amount of aneuploidies in the embryo not allow that arrive to blastocyst stage. And in that case, depends of the focusing of the patient. And if you try to convince to the patient to move to egg donation, sometimes it's good idea to have results and report in the hand. Otherwise, the patient try again and again and again and spend a lot of money and time, and maybe arrive 2 years later to egg donation. And then no makes sense, okay? And the idea is, in our experience, comparing day 3 to day 5, okay? And it's very, this moment, I think this, the experience in 2 years ago, the percentage of abnormal embryos at day 3 is 80% comparing to 64% at blastocyst stage. And if you can see here, is really impressive how the chaotic pattern decrease when you perform the biopsy at blastocyst stage. And this is really is logic that this happens because most of the chaotic pattern embryos and complex aneuploidy embryos not arrive to blastocyst stage. And if you have very good culture condition, you have a natural selection in your lab. And the advantage to perform blastocyst biopsy is also you have less embryos for biopsy, you have less work in the lab, and then it's cheaper for the patient. Pay less money for the analysis. At the end, it's the same because all the abnormal embryos that they have on day 3 will be have in day 5, okay? And the normal embryo arrives to blastocyst stage. And you can see here the behavior of how the the percentage of abnormal embryos increase with the age, independent if you perform the biopsy on day 3 or day 5, okay? The increasing is also for day 5. And in our experience, performing day 3 is really the same that performing day 5. And to demonstrate that, we perform the biopsy on day 3, and the abnormal, and those embryos that was diagnosed as abnormal and arrived to blastocyst stage, we rebiopsy this blastocyst, we reanalyze, and in 98% of the cases, the diagnosis was confirmed. And also, we fixed the complete blastocyst and analyzed by FISH using some proof for any of aneuploidies in the embryo. And performing day 5 blastocyst, we perform the biopsy at blastocyst and then perform the analysis. And some blastocysts, those blastocysts, abnormal blastocysts, was rebiopsy, and the concordance rate was nearly to 97%. In the case of young patient, young than 30 years old, you can see here performing day 3 biopsy comparing to day 5 biopsy. There is not any difference in the clinical parameters. Pregnancy rate per transfer, implantation rate, and pregnancy rate per cycle are identical performing biopsy at day 3 and performing biopsy at day 5. And finally, I would like to mention that nowadays PGD refers only for monogenetic disease or single gene disorder. And this is the original purpose of PGD at the beginning. And this is the idea is offer a patient, a boy transmitting inherit condition to their children and potentially eliminate from their family. And through PGD, we can identify if the embryos are affected for a specific single gene mutation linked to serious disease. And another approach that we can apply is combine PGD plus PGS. And then we can transfer completely normal embryos for the disease and completely free for aneuploidies, okay? And in this way, you can improve the clinical outcomes. Maybe in 10 points, combine both analyses. In conclusion, we can perform the PGS using array technology or NGS. And in our experience, day 3 or growth factor biopsy offered similar result in similar good prognosis population, and probably for advanced maternal age and low ovarian responder, day 3 could be more informative. And really we can perform this kind of analysis in patients in couple with high aneuploidy risk, advanced maternal age, recurrent miscarriage, recurrent implantation failure, male factor, and previous trisomy pregnancy. And even for young prognosis patients, get advantage for this analysis, and just in order to focus in the only purpose to make a single embryo transfer, avoid multiple gestation, and also you can combine PGD and PGS. We like to thank our team in Igenomix that implement the PGD program and PGS program. We are pioneers group in Spain and Europe, and nowadays we cover the world offering these kind of services. And thank you so much for your attention.